Hello, guys. Good day. And here I am again, Miss Merle, to have the topic nucleic acid amplification. So, like PCR, it is a vital molecular biology technique which creates numerous copies of specific DNA or RNA, and it's widely used in research, diagnostics, and forensics. And it involves temperature-dependent reactions amplifying a target sequence. And then this process is driven by DNA or RNA templates. We have primers, polymerase enzymes, specifically the DNA polymerase, and nucleotides, and it results in an exponential increase in copies. So that's why we have the word amplification, or there is this thing known as an exponential increase. So this technique is a pivotal in medical diagnostics, genetic research, forensics, and it has really revolutionized molecular biology, guys. So for our outline, amplification or in having the copies in exponential increase, we can do that through target amplification, probe, and signal amplification. And under target amplification, we got PCR, and we also have transcription-based amplification systems. And under probe amplification, we have ligase chain reaction and strand displacement amplification. And for our signal amplification, we can utilize branch chain DNA or branch DNA amplification, hybrid capture assays, cleavage-based amplification, and cycling probe. So for our objectives, we have the following. So we need to compare in vitro nucleic acid amplification assays like PCR, branch DNA amplification, ligase chain reaction, transcription-mediated amplification, and Q replicase. So we will be comparing them in terms of target nucleic acid, procedure elements, amplicon types, employed enzymes, and applications. And we need also to know how to explain PCR modifications, or what are the types of PCR. What are those? And then we need to discuss how amplicon detection in various amplification methods, and also how to design PCR primers for a given target sequence, and how to differentiate between target and signal amplification. So, a while, guys. Let's just okay this one. So, early nucleic acid analysis, guys, were constrained by limited material, and producing ample copies of gene sequences required labor-intensive methods like cell culture. In Bangkok, I'm doing a lot of cell cultures, guys. And then, or isolation of large genomic DNA amounts. Cloning allowed multiple copies in bacterial plasmids, and it was tedious, tiresome, and certain sequences resisted propagation. So the advent of specific DNA sequence amplification, notably through PCR, revolutionized nucleic level analysis, and subsequent methods, often modifications of PCR, allowed or enabled versatile DNA analysis. So amplification techniques can be categorized based on whether they amplify the target nucleic acid, a specific probe, or the signal used for detection. So for target amplification, it involves creating millions of copies of a specific nucleic acid sequence for in vitro detection. So this is akin to or similar to cell culture growth for visualizing cells on an agar plate, but target amplification is faster and taking hours to days. So PCR was the first and the prototype method, and is commonly used for this purpose. So PCR started with Carrie Mullis, and in 1983, Carrie Mullis conceived PCR. So a method of amplifying DNA in vitro, and by doubling a target through repeated cycles, he could achieve million copies. And despite initial challenges, the first successful amplification was a fragment of E. coli. plasmid. And 2 years later, uh, PCR's practical application for diagnosing sickle cell anemia was published, and originally called polymerase catalyzed chain reaction, it quickly became known as PCR, guys, shortened to PCR. And then since then, it has evolved to be user-friendly, automated, and widely applicable in clinical labs. So that's with our So for the basic procedure, when the cell replicates its DNA, so in the process of DNA replication within cells, the existing double-stranded DNA serves as the template. So the starting point of our PCR is our template, and it's constituted by our double-stranded DNA because that dsDNA serves as a template, so it provides the sequence for nucleotide bases like our adenine, our thymine, cytosine, and guanine. And so DNA polymerase catalyzes the addition of nucleotides to the growing strand, guys, guided by a primer because the primers will there's this hybridization or annealing in a complementary basis. So they will just not just attach like if they would want to or not, but they're really base pairs. So primers will guide the our how can I say that? The primers are really significant when it comes to the sensitivity of our PCR. So that's with the primer, and PCR mimics in vivo DNA replication. So it's just like we're doing DNA replication but in vitro. See, because we cannot do it in vivo inside living organisms like humans. So we can do that easily in the lab through this technique. So we have DNA synthesis or replication. So utilizing the same components, guys, to produce two copies of double-stranded DNA from one within 1 to 2 hours. So somehow, if I'm trying to summarize things here. So guys, if you will ask me if how long can we finish a PCR reaction, usually it's an hour or 2 hours. And we can say that this is really rapid as compared to cell culture, guys. So this rapid amplification is advantageous compared to the days it would take for a cell to achieve the same in vivo, and PCR excels at amplifying specific targets. PCR is really specific. That's why this is our confirmatory test for HIV/AIDS. And similar to, we also have southern blots. Have you noticed we have southern blots, western blots, northern blots? So, um, western blood is for HIV, right? Have you remember that? Southern blood is for DNA, northern bloods for RNAs. So, what I have said earlier. So this is similar to southern blood's ability to analyze specific regions in a complex background, and the amplified target can undergo numerous analytical procedures. So we have downstream application. That's what we call the analytical procedures that will come right after we have extracted or we're able to analyze. We're able to resolve and detect our target sequence. Then our target sequence can further undergo downstream analytical procedures. And PCR components, guys, include the DNA templates. Again, that double-stranded DNA that serves as a template. We have the primers, we have nucleotides. Of course, primers, we have the forward and the reverse primers. We have nucleotides, the DNTPs. We also have our polymerase and buffers, and they all undergo an amplification program with specific or specified cycles and steps. And these cycles, these cycles, guys, involve holding samples at specific temperatures for designated times with temperature changes influencing the reaction. So it's really the temperature that will determine the reaction that happens. So that's why, as what you have noticed, we have three steps for one cycle of a PCR. So we have denaturation, we have annealing, and we have extension or elongation. And they have different for every step, the temperature varies. Like, take for example, during denaturation, we have 90 to 96. For annealing, we have 50 to 70. And for the extension, we have 68 to 75. And also, the time has to be, sorry guys, the time also has to be changed. Okay, so again, the temperature changes influences the reaction, and a common three-step PCR includes, again, we have denaturation, where what happens with our denaturation. So denaturation, guys, sorry, it is where our double-stranded DNA separates into single strands to be replicated. So if we are talking about a denatured DNA, so meaning the double strands are now single strands, okay? So this is where our double-stranded DNA separates into single strands to be replicated. It cannot replicate double-stranded DNA, okay? So that's why we really have to start with denaturation. That's the primary or the initial step of our PCR cycle. So the initial denaturation uh, step is extended for genomic or large DNA templates, while subsequent denaturation can be shorter. So see here, subsequent denaturations can be shorter. So the next and most critical step for the specificity of our PCR, guys, is our annealing step. So that's the most important step for our PCR. Annealing, so hybridization, there is attachment. So although it is the second step, so you might ask me, during denaturation, we're just changing the temperature so that there is melting, there is breakage of our double-stranded into single-stranded DNA. However, during our annealing, guys, in the second step of the PCR, so the two oligonucleotides that will prime the synthesis of DNA anneal. So anneal, it's also known as to hybridize, or in layman's term, it's easy to understand, to bind or to attach to their complementary sequences on the template. So take for example this one. So the primers are complementary to they have sequences that are also complementary in the template DNA, and it is, guys, you might ask me, Miss, shall we amplify the entire DNA template? No, it's just there's this target sequence that we need to amplify, or the region under investigation that we need to amplify. Okay, so I hope you get the point with this one. So it's not the entire length of our DNA template that has to be amplified, that has to be, that we need to create a million copies, but it's just this region under investigation, or our target sequence, that has to be amplified, guys. And then again, the primers dictate the part of the template that will be amplified. So what really governs the specificity of our PCR, it's because of our primers, and a good primer design is really an should be an utmost consideration if we are about to perform a PCR uh, technique in the laboratory. And then, in other words, the primers determine the specificity of the amplification. So, in short, why is PCR, why is this methodology highly specific? It is because of the presence of primers, and primers tend to anneal or hybridize two complementary sequences on the DNA template. And then, again, as what I have said, it is really essential that the temperature for hybridization has to be in optimum levels with the primers and reaction conditions. You need to remember that primers must have the same melting points. So annealing temperatures will range from 50 to 70. So some books there is a slight variation, but as long as it's within the range, it's okay and are usually established empirically. Okay. And then a starting point can be determined using the melting temperature of the primer sequences. So you will learn this in our other lecture, how to determine the melting temperature of our primer sequences. It's based on hybridization and stringency. And so reaction conditions, salt concentration, because guys, you also have to consider the salt concentration in your reaction. But like, ask me how. I hope I can really find the time to teach you in using the software for primer design. So you will know how to perform all these, how to optimize, how to set all these so that you will have a good primer to start with, the forward and reverse. And then we also have secondary structures. Secondary structures, guys, RNAs and DNAs tend to fold within themselves. So it's inherent or innate in their, it's an innate characteristics to them, to them. So we need to avoid secondary structure and the following because they can really change in fact, even the length of our primers can affect our annealing or hybridization. So it could be that this length you need a higher temperature, or if it's shorter, then also the temperature of our PCR cycle is affected. So before moving, I guess it's good to touch, you really have to memorize what constitutes, what are the components of our typical PCR reaction. So always we will start with the DNA template. Okay, so that's the sample DNA, and our sample DNA, that's the DNA that we got from our DNA extraction. That's the very first thing, followed by we tried to resolve that, we tried to detect that in our, commonly in our agarose gel electrophoresis, and you wanted to quantify, we can cut the gels where the bands are present, and then we will do downstream applications to test. So it's just like that, guys. So again, we've talked too much. We have the DNA template. And then always remember, if we've got that template, we also have the primers. Please do not confuse with the terminologies here. So the DNA template that holds our target sequence. It holds the region that we wanted to investigate. While the primers, they are playing a very important or essential role in directing DNA replication or synthesis to the desired region. Why? Because they are the ones that will again anneal or hybridize to the complementary sequences present on the template. And then we also have the DNTPs. So we have the DNTPs, or deoxynucleotides, in the form of, we have DATP, DCTP, DG, and DTTP. So the primer, guys, cannot extend the DNA sequence without using. So these are the things, these are the stuffs that are being added once there is an exponential increase, right? So you might wonder, where shall they get the building blocks to extend the primers? What component responsible for extending the primer is our DNA polymerase. So it's really a chain reaction. It started with the template, and out from the template that we have got primers that will bind to the uh, complementary sequences present in the template, and then once the primer is there, the polymerase will find it, and then it will extend the primer by adding DNTPs. These are the DNTPs. These are the building blocks for the growing DNA strand. And to stabilize, to obtain an optimal pH, no, yeah, true. to have a good pH, and also for so that we can have an optimal annealing with our primers and that of the template. We need to have the presence of our cations. So we need to have the potassium chloride or magnesium chloride. So monovalent cation for optimal hybridization of primers to the template, and we have the divalent cation, which is this one is required by our polymerase. So the polymerase is also dependent on MgCl. And then the most common buffer to maintain the optimum pH of our enzymatic reaction, because this is driven by DNA polymerase, we need to have the tris buffers, and specifically with a pH of 8.4. So guys, please memorize this table, okay? So table 7.1. Actually, if you can memorize this, you can really have a rough understanding on everything. Okay. And next, we have the elements of a PCR cycle. So one cycle we have denaturation, annealing, and elongation or extension. That's one cycle. Okay. So how many cycles usually do we have for PCR? Like it's 34 to 50 commonly. So most of the labs here in our country, we have 34 cycles, 36, 38. So it's up to the institution. So we're done with denaturation. Again, there's melting, annealing, hybridization, annealing, primers, remember, and then extension. What's responsible for extending this one? DNA synthesis can happen because, please don't forget our table. See? So the polymerase enzyme that extends the primers adds DNTPs. During extension, DNTPs, these species are added. Okay? Okay. And then the third and the last step of the PCR cycle is the primer extension step. Okay, so essentially, this is similar to DNA replication or DNA synthesis, as what I have mentioned in the past few minutes. So in this step, our polymerase synthesizes a copy of the template DNA by adding again, so the addition of the NTPs or nucleotides to our already hybridized primers, because these primers were able to anneal already. And then DNA polymerase catalyzes the formation of the phosphodiester bond between an incoming DNTP determined by hydrogen bonding, blah 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 blah. So, Miss, what's the important thing that we should remember here? So, DNA polymerase replicates the template DNA by simultaneously extending the primers in both strands of the template, and this is really dependent on the enzyme's optimal temperature, because we cannot just choose any temperature we would want to have. DNA polymerase is really specific and will function best. Optimum temperature is at 60 to 72. Some books would say 68 to 75. So it's up to that. Still within the range though. And in some cases, the annealing temperature is close enough to the extension temperature. That's why we have the two-step PCR as opposed to the three-step. But this one is not so common. So the most common is the three-step PCR. So at the end of the three steps or one cycle, see? So one cycle is composed of three steps. And after one cycle, we can have one copy of the double-stranded DNA that has been replicated in two copies. So as we increase the temperature back to 90 to 96, so another cycle starts. So that's with PCR. That's why it's a chain reaction. It keeps on creating cycles. How many cycles again? We've got usually 34 to like 50. And then guys, see, this denaturation of the DNA target used to be a double-stranded, and then it's already separated into single strands. And always remember, guys, if we are adding primers to our PCR master mix, the primers has to be in a very vast excess. And in the second step, this is just a review, the annealing. So our primers will bind or hybridize. So the primers hybridize to their complementary sequences on each strand of the denatured template, and the primers are designed to hybridize to the sequences flanking the region of interest. So what do you mean by flanking the region of interest? Meaning at the sides, left and right of our target sequence. So our primers will attach at the sides of our, not directly to our target sequence, but they will flank the region of interest. Okay, and sa kilid na siya in short. Ara sila mo hybridize sa kilid, mo mean sa flanks, di ba? Sa kilid man ang beside at the side, okay? And then components of PCR. So since this one is an in vitro DNA synthesis, so we need to make sure that all of the components needed are there and are combined in the best of conditions for DNA replication to happen in vitro. So what are those? What are the components we need to? Of course, the starting point, the template to be copied, our DNA template, our primers. For prime synthesis of the template, we have our DNTPs or nucleotides, the 4 nucleotides. We have our very important DNA polymerase enzyme, which is responsible for adding our nucleotides and buffers to maintain an optimal pH. So, and we also have the monovalent and divalent cation. So this one, you can see this as in a summarized form with table 7.1, the components of a typical PCR reaction. Okay. So guys, we have the primers. So if we're talking about primers, guys, they are really critical components of our components of the PCR because they are responsible for determining how specific our PCR reaction is. And our primers are similar to probes in our northern blots, southern blots, and hybridization procedures. And they are chemically manufactured in a DNA synthesizer. So chemically manufactured in a DNA synthesizer. But the thing is we can actually set the specs of our primers. We can set it online and we can give the thing that we wanted to have, the length of our primers and what are the sequences that they are supposed to bind. We can do that, and then the manufacturing company will just provide us with the primers. Actually, we can buy it, but we need to specify what are the characteristics of our primers to the manufacturing company. And then again, primers are really intended or designed to have sequences homologous to the size flanking the region to be analyzed. So if this is our target region, then our primers, they have sequences that are homologous to the sites flanking. So what are those sites flanking? Ang sites nga naa sa kilid sa target region. Naon pagsabot ba? And primers are, guys, unlike our template that started as a double-stranded DNA, our primers are single-stranded DNA fragments. And remember, they are having this set number of bases. So should only be 20 to 30 in length. So the forward primer will bind to our target sequence or target DNA sequence just 5' to the sequences intended to be amplified, and our reverse primer must bind, just 5 prime to the sequences to be amplified on the opposite strand of the DNA. Hence, the design of primers requires really knowledge, guys, of the target sequence. Because if we don't know what sequence we're looking for, it's just like finding a needle in the haystack. So, we really, Miss, how can we know our target sequence? For example, you're looking for the thalassemia gene. So in the software, you'll just encode thalassemia, and then that will set everything in motion. Like you'll just fill up a lot of boxes there in the software. Yeah, I think it's a good thing if we really have that thing known as primer design in our laboratory. I think it's not present in our present lab manual. So sorry guys, but we're still also are learning. We teachers are still learning what are the things we need to put in. I'm not the primary author of the manual. It's the dean. So yeah, that's why the primary design was not incorporated in our lab manual. So going back, the placement of the primers will also dictate the size of our amplified product, and binding of primers is subject to the same physical limitations as probes. So the primer sequence and length, this is what I have said. The sequences present in our primer and how long will affect our optimal conditions in which our primer will bind to its target. So Miss, again, do we have what are the things that we need to consider? So we need to consider the melting temperatures, and then we can calculate it by using the equation for short DNA fragments described. So you will be calculating that one too, our melting temperatures, and you will learn that more on our chapter 6 discussion. And then what else? So our the setting the optimal annealing temperature, the critical step for the specificity of amplification reaction. So guys, primer design number one that you should consider is the TM, or the melting temperature. So that, and also consider that our forward and reverse primers must have the same. You should set them at the same annealing temperatures so that they can function in the same temperature. So they can work in unison, right? And they can be adjusted by increasing the length of the primers or by placing the primers in areas where what? With more or fewer G's and C's in the guanine and cytosine in the template. So guys, we also have the thing you need to remember here, primer dimers. So the bad thing, though primers govern specificity for our PC reaction, but they also tend to bind into themselves. That's what we call primer dimers. We have intra and inter primer binding. So before going to this one, let's have this. So the nature of primer structure determines the accuracy of binding. See, and then we also have, but we can also encounter problems like mispriming, that it will bind to sequences in which those sequences are not the sequences that we're looking for. See, a fragment synthesized by mispriming will carry the primer sequence and become a target for subsequent runs of amplification, giving us a false positive result. Though actually, it's not supposed to amplify that because it's not the sequence that we're looking for. But there is this aberrant primer binding or mispriming. And then what's the disadvantage? Our products which are misprimed, the amplicons which are misprimed will consume our components away from our intended reaction. So what's the result? Of course, there's interference with our result interpretation. See, false positive. And then secondary structure, see, DNA internal folding and hybridization. So DNA strands also tend to fold into themselves. And this can also interfere with our PCR technique. So primer sequences that have internal homologies, especially at the 3' end. So primer sequences that have homology, especially at the 3' end. So this is the generation like at the 3' end, forward reverse. And then take note that, for example, these bases are complementary to each other. Like, for example, this one is A to T, C to G, C to G, C. They are complementary. And what's the problem? They tend to hybridize. They tend to anneal. So it's not a good thing. This will result to primer dimers. So, and the primers will be extended by the polymerase using the opposite primer as the template. So, what happens to the product? So, the product denatured in the next cycle and will compete for primers with the intended template. So, again, primer dimers are that nice to see. So there are PCR products that are just double the size of the primers, and they result in binding of primers onto each other through short 2 to 3 base homologies at their 3' ends, the copying of each primer sequence, quite lengthy, sorry. So the resulting doublet is then a very efficient target for subsequent amplification, and the entire primer sequence does not have to bind to the template to prime synthesis. However, the 3' nucleotide position is critical for extension of the primer. So that's the thing. So again, guys, we have aberrantly binded primers, aberrant primer binding, mispriming. We have primer dimers, and we also have tailed primers. So tailed primers are they are made, they are designed to alter sequences to one or both ends of the PCR product. So these primers are added or to alter one or both ends of our amplicons. So we're done with our DNA template. We're done with our primers. Now, no, let's go back to our DNA template. So our template, it doesn't have to be really always double-stranded. We can also have a template which is single-stranded. And usually in the laboratory clinical setting or in the clinical sample. So our template, we can get it from the patient's genomic or mitochondrial DNA or from microbes and parasites that might be infecting the patient. So genomic DNA will have only one or two copies per cell equivalent of single copy genes to serve as amplification targets. So again, what do you mean by that? Like, we cannot have that much of the copies per cell equivalent. So often we only have one or two copies. See? And that's not much. That's why we need to amplify. We need to amplify our targets. We need to amplify the targets present in our DNA templates. So with a robust PCR reagents and condition, we can get as much as nanogram amounts of genomic DNA. This is sufficient for us to give reliable and consistent results. So for us to perform clinical analysis, it's best to have 100 ng to 100 micrograms of DNA. Usually, this is the amount that we need, that we can derive enough sequences. So again, don't forget this one, clinical analysis, 100 ng to 1 micrograms of DNA is usually used. And then, however, if were able to purify and we're already, we had performed cloning already of our DNA. So from a previous amplification, so lesser amounts are required. So, Miss, what templates can we consider as the best templates? So, those templates should be in good condition. They should not contain proteins that can contaminate, and they should be free from breakage or without nicks, so that because the breakages or nicks can really terminate DNA synthesis or cause misincorporation of nucleotide bases. So templates with high GC content and secondary structure may prove more difficult to optimize for amplification. So again, templates having, again, what do you mean by the secondary structure? When the protein, no, no, when the DNA then the DNA strands tend to fold into themselves. And then if they have high GC content, so the DNA region affected by, this is an example like Fragile X syndrome. The region, this FMR1 gene is having so much of GCs and it's not good. And then after our DNA template, we have our deoxyribonucleotide bases, our DNTPs, our nucleotide triphosphates. So during our replication, during synthesis, these are of course required. That's why we need to supply them in our PCR master mix. And we have our 4-deoxynucleotide triphosphates, our DNTPs, adenine, thymine, guanine, and we have our cytosine. So we need to add it in our PCR master mix in our Eppendorf tube, of course. And for the NTP concentrations should be higher than the estimated km of each DNTP. So standard procedures need to have zero point 0.1 and zero point 0.5 millimolar concentrations of each. And then we also have the Diaza GTP. So this one is usually this one is required for if we wanted to have a very optimized test, that's the time we need to have this DNTP. And then for our DNA polymerase, so as what you had in our laboratory, those videos you have seen, Yellowstone National Park, wherein we can isolate our Thermus aquaticus, that thermophilic bacterium. So that's the source of our DNA polymerase. So our automation of our PCR product was greatly facilitated by the discovery of our thermostable enzyme. Thermostable enzyme, it's heat labile. No, it's not heat labile, sorry. It's highly stable in very hot conditions. They can grow at temperatures above 70 degrees Celsius. Hence, we can say thermostable or thermophilic, and they are isolated in thermal springs. Okay? So it was when Karimos first performed PCR, he was sadly, he haven't, he haven't discovered Thermus aquaticus yet. So he used E. coli. However, every time that every step of the PCR, there's a need for him to add, because this polymerase coming or isolated from the E. coli is easily denatured in high temperatures. So it's, how to say that, more of a hassle, right? And labor intensive. But right now that we have already our attack polymerase from our thermophilic, from our thermostable bacterium, Thermus aquaticus. And our Thermus aquaticus, guys, it has little or no 3' exonuclease activity. That's why it's also chosen. And using an enzyme derived from a thermophilic bacterium meant that the DNA polymerase could be added once at the beginning of the procedure and it would maintain its activity all throughout the heating and cooling cycles. So that's really the advantage of using a thermostable enzyme, Taq polymerase, coming from our thermophilic bacterium Thermus aquaticus. And we also have other alternatives for our Taq polymerase. So we have our TTH polymerase from our Thermus thermophilus. We can also use TTH polymerase, not our Taq, but our TTH polymerase. in our reverse transcriptase PCR. So this one for our RNA, for our RNA uh, determinations, and where the starting materials and RNA template. See, so the addition of proofreading enzymes like Van polymerase allows our TAC or TTH to generate products over 30,000 bases in length. And we also have the, how to say, our Stoffel fragment, which lacks the N terminal to 89 amino acids of Taq polymerase and its inherent 3' 5' exonuclease activity. So the Stoffel's fragment at high temperatures half-life is about twice that of our Taq polymerase, and it has a broader range of optimal magnesium chloride concentration. So if we are doing allele-specific PCR, then our Stoffel fragment is the best for that. So much for, let's go to our PCR buffers. So PCR buffers give optimal conditions for enzyme activity. So we have potassium chloride and we also have our magnesium chloride. See? So magnesium chloride, guys, is an important, it's a monovalent cation and it's really good in buffering. It's a good buffering component. And these are present as salts and they affect the denaturation and annealing temperatures of the DNA and the enzyme activity. So an increase in salt concentration makes longer DNA products denature more slowly. So please highlight this part. Guys, so an increase in salt concentration, meaning there's slow denaturation of our DNA product. So there's a preference of amplification then for shorter molecules. So it's better to have shorter molecules because they are preferentially amplified. Okay. So the influence of buffer and salt conditions varies with different primers and templates. We also have magnesium chloride also affects primer annealing and is very important for enzyme activity because uh, what's the rule for that? A decrease in magnesium ions lowers enzyme efficiency, and there's a low PCR product yield, or there is low amplicon yield. While for if we have an increase in magnesium ions, so it it's not a nice scenario because it increases mispriming. So our yields will tend to be non-specific. So again, for our, if we have an increased salt concentration here in our potassium chloride, it slows the denaturation of our DNA product or amplicons. So that's why it's better to have shorter molecules. And then for under our magnesium chloride, so a decrease in our magnesium ions. So there is lower enzymatic efficiency. So there is decrease, or we have low yields of our PCR products or amplicons. But if we have an increase of our magnesium ions, it's not also a good thing because it promotes mispriming, and mispriming we have a yield, though, yes, present, but the products or amplicons are non-specific in nature, guys. So are we clear with that? Then what else? So trace buffer and our accessory buffer components, they're also needed, and they maintain the pH of the buffer usually from between 8 and 9.5. We also have for our accessory components for our to optimize our PCR reaction. We have, what are these accessory components? So we have bovine serum albumin. I'm always using this in Bangkok. We have dithiotritol. We have formamide. We also have the chaotropic agents. Chaotropic agents in the form of Triton X. I'm also using this. We have glycerol and DMSO. Yeah, I'm also using dimethyl sulfoxide or DMSO. So going back to our where is that? BSA or our bovine serum albumin derived from bovis, so cows. So this one offers enzymatic stability by binding to inhibitors, while our diethiothretol, so it provides reducing conditions which enhances enzymatic activity. We also have formamide, where is our formamide. So formamide, it lowers the denaturing temperature of DNA with increase in the secondary structures. So there's an increased availability for primer binding. That's for formamide. So you really have to know their functions too, guys. That's for our P1 exam. And then for our what are chaotropic agents? There's chaos, chaotropic. So uh, they how can I simplify chaotropic? So they disrupt the hydrogen bonds between water molecules. So they are more on disrupting molecular structures. That's why chaotropic. So they are solutes that can disrupt hydrogen bonding. So that's the easier thing for us to remember. And they tend to decrease secondary structures of DNA, so allowing polymerase extension through difficult areas. So somehow they're not so bad at all. See, because they reduce secondary structure to allow polymerase extension through difficult areas. So that's why our they can also give stability to our enzymes. So those are our accessory components. The accessory components were not mentioned earlier in the components under our PCR reaction because these are just added to arrive or to achieve better PCR reactions at optimized levels. You can not add them, but if you will add them, they will give you something out from the ordinary. So that's with our accessory components. Okay, guys. And what else? So here, buffers, open the buffers. So the enzyme target and primers are then added when necessary. So, Miss, what do you call the collection of everything that we only need to add the template or the primers? It's known as our DNA or RNA master mix. So it's our master mix, guys, or MM. And next, let's have our thermal cyclers. Thermal cyclers, guys, the first PCRs were performed using multiple water baths or heat blocks. So, like manually, you will move the tubes from one block to another or from one water bath to another. But that's an open system in the past. And see, the tubes were moved from one temperature to another by hand. So, for me, it's prone to contamination. It's prone to denaturation and unwanted denaturation. So, the good thing right now, we're able to automate PCR, and why we are no longer performing this tiresome thing of transferring the microcentrifuge tubes from one water bath to another water bath to another water bath. Now we have our thermal cyclers or thermocyclers, and they are really designed to give a rapid and automatic ramp or change of temperatures, like from again, if for during denaturation we have what's the denaturation temperature from 90 to 96, and then if it can after 20 to 60 seconds it can change to 50 to 70, staying for 20 to 90 seconds, and then it will have 68 to 75 for the extension for 10 to 60 seconds. So see that rapid change of temperature, guys, is temperatures. This offered by our thermal cyclers. Actually, our thermal cyclers are not radioactive. So there's no radiation there. You should not be afraid of radiation, but the reason why we need to stay away from our cyclers if it's cycling because we wanted to maintain the temperature, around it like 3 meters to be really stable. Because if you will keep on passing, like you wanted to get something near it and put something in the ref and do something like that, this for me it can really affect our thermal cycler, how it cycles the temperature. So it's better to stay outside from our molecular lab if we're using our thermal cyclers, our biorad thermal cycler. So the earlier or the versions of thermal cyclers in the past were designed as heaters or coolers. And they also use something like wax or oil to prevent condensation of the samples on the tops of the tubes during temperature changes. But it's really difficult. Handling such things is difficult, and that's why the recent cycler models were designed with heated lids. So that's the one we're having. We have heated lids that can really eliminate, or we can remove the need for us to use wax or oil vapor barriers. And then there are numerous manufacturers of thermal cyclers, and you might ask me, we have Birad, and they have differences when it comes to their programming, how they program their softwares, but still they follow a common mm, diagram or flowchart. And samples may be held in open chambers for air heating and cooling, or in sample blocks designed to accommodate 200 microliter tubes or zero point 0.2 ml tubes, usually in 96 well format. So it's just the same with what we have in the lab. And some models have interchangeable blocks to accommodate amplification in different sizes. So those are the modifications. And we also have our real-time PCRs. Our real-time PCR is like you're trying to detect amplification cycles as it happens in real time, and why it's being able to measure amplification, and why it can determine the number of cycles running or being performed. It's because it is equipped with detectors, fluorescent detectors, to measure PCR products. Because that's why we also have probe amplification. So we can attach probes to our templates, DNA templates. So those probes attached will be detected by the fluorescent detectors present in our real-time PCR. So that's the thing. And then for routine PCR in the lab, an appropriate amount of DNA that has been isolated from a test specimen is mixed with other PCR components. So that's what we call our pre-PCR. So pre-PCR work. And we're supposed to have a designated place or area that is clean and free of amplified products. So pre-PCR, that's the first, supposedly our first table. And the last table should be our post-PCR. So the dirtiest area is the step or part in which we are amplifying the DNA. Okay, so that's the thing with this page. Next. So here. So guys, see, the technology starts the run and walks away until it is complete. So you can just leave the thermal cycler running for again 1 to 2 hours. And then that's why our lab is 3 hours. So after the PCR, we have a variety of methods that are used to analyze our amplicons or our PCR product. And the most common is our gel or capillary electrophoresis. This one is our agarose gel, I think. Yeah, this is an agarose gel. So the amplicons are resolved using an agarose gel. So Miss, what is the first lane, the first column, number 1? So this represents the molecular weight markers. So you'll know because if these ones closer to the cathode are large molecular weight entities, while these closer to the anode are small and negatively charged. They are near the top. They are positively charged and they are large molecules. See, guys, there's an example of mispriming here. It's non-specific. We also have like the ghosts, the primer dimers. So we have our marker here, but it's not really in line, and they are not intended to be detected because our PCR product is just here, and usually the dimers are quite below the PCR products. And just same with the other testing in the laboratory, guys. We have a lot of controls for our PCRs. Sabi niyo, wala ni controls. Daghan ni siya controls. We have the positive control. We have the negative control, contamination control, reagent blank. We also have another negative control, the negative template control. And we also have the internal control, the amplification control. So a lot of controls, guys, right? So as with any testing or diagnostic assay, running the correct controls in PCR is important for maintaining and ensuring the accuracy, of course. And then, so again, what are those controls, miss? So positive control. So the positive control will, it ensures that the enzyme is active, that the buffer is at its optimal level, primers are priming with the right sequences, and the thermal cycler is cycling. And then that's our positive control. So we need to have our positive control. We also have two sets of negative controls. The first one is the negative control that is also known as our contamination control or reagent control or reagent blank, sorry. So again, the negative control for contamination control or reagent blank. So quite a lot of controls here. So that one, it ensures that the reaction mix is not contaminated with DNA template or other amplified products from the previous run, because the most common reason of contamination are the amplicons from the previous run. So for us to eliminate the doubt on that, we will use our negative control without DNA. So that's known as our uh, reagent blank or contamination control. We also have the negative control with DNA. So negative template control because it lacks the target sequence. It lacks the target sequence, and this ensures or guarantees that the primers are not annealing to sorry, that the primers are not annealing to unintended sequences. So that's the thing for that. We also have that control known as the internal control or our, where is that? The internal control or our amplification control. See this one, amplification control. So internal control and internal control. Okay. So the addition of second set of primers and an unrelated target. So is done to show that the reaction is working even if the sample is not amplified. So somehow it's very important because you will know that your master mix is working, that things are working in the right direction if the test sample, even if the test sample is not amplified. So this amplification control is best for PCRs to be reported qualitatively so that we can prevent the problem on false negatives. So I'll stop because it's already an hour, and I'll do another, yeah, we'll do another set for the discussion. Thank you and good day.